Welcome everybody to this Ask the Expert session. On the panel are Dan, Duncan, and myself, and we are all lecturers in the School of Life, Health, and Chemical Science at the Open University. And all of us work in the cardiovascular field. So what can you expect from this session? First of all, I'm going to talk about the structure and the function of the heart. After that, Duncan will do a live demo of technologies you can use to measure heart rate and explain how heart rates are regulated. And finally, Dan will talk about heart diseases and the broken heart syndrome. So what does a heart really look like? On the left-hand side, you've got an image of a human heart, and you can see that the heart doesn't quite look like the diagrams or the pencil drawings we do on paper, but it has more the shape of a fist. In fact, the heart of an adult human being also has about the size of a fist, and it weighs around 300 grams. What you can see here are blood vessels that are supplying the heart itself with blood. And you can see these yellowish areas, which are areas of fat that surrounds the heart. And this fat is not only harmful, but the fat is also protective. So not all fat is bad fat in case of the heart. On the right-hand side, you see a schematic diagram of the heart, where you've got two smaller chambers here on the top, which are called atria. Then you've got the two main chambers of the heart, which are called the ventricles. And these have a very thick muscular wall that aids with the contraction of the heart. To contract, the heart gets electrical stimuli. And these start off at the SA node or sinoatrial node, which is located on the right atrium. And that's the heart's pacemaker and causes regular electric signals, which spread across the atria and to another pacemaker region, which is called the atrioventricular node or AV node, which is found where the atria and ventricles connect with each other. So the electrical excitation then spreads via the ventricles and along the ventricles. And this electric signal causes the muscle cells to contract and causes the heart to rhythmically beat. So you've seen the static heart now. Let's go and look at a beating heart. At the Open University, we've developed an augmented reality heart. And I think the link for accessing this heart is going to be put into the chat box. And I'm going to open this app now. So what you can see is a three-dimensional heart, and you can move the heart around, look at it from the different sides. So you can, for example, see here the atria, which receives the blood from the body and from the lungs. You can open the heart, and then you can look inside the ventricles. So here you've got the two main chambers of the heart with a thick muscle wall. And in this white silvery color, you see the nerves that transmit the electrical conduction. And here you see some valves between the atria and the ventricles. What we can do with this app is play the heartbeat. So you can see the contractions of the heart, and the contractions start first in the atria, and then the ventricles connect. And we can show the blood flow. So we see in blue deoxygenated blood, which has a low oxygen content. It comes from the body into the right atrium, into the right ventricle, then leaves the blood here, goes into the lungs. In the lungs, the blood takes up oxygen, returns via the left atrium into the left ventricle, and then it is moved via the aorta into the body and distributed throughout the whole body to deliver oxygen to all the organs and tissues in the body. So after you've seen how the heart contracts and works, I'm going to go back to our presentation. And ask the question, you've seen that the heart is a muscle, it's contracting regularly throughout your whole lifetime. So does it only contain muscle cells? And we call the muscle cells of the heart cardiomyocytes. And there's two answers to it. If you look at the volume of the heart, yes, the majority of the heart is made from cardiac muscle cells. 70 to 85% of the heart's volume is made from these specialized heart muscle cells. But these cells are really, really big. So if you look at the number of the cells, you can see that actually the heart contains several other cell types. For example, fibroblasts, endothelial cells, smooth muscle cells, and adipocytes, which are fat cells. So the heart also has connective tissue, it has blood cells. To perform its function, the heart needs a lot more cell types than just cardiomyocytes. But if you look at the volume, the heart is made mostly of muscle cells. And how do these cardiomyocytes look like if you look at them at a microscope? So here on the left-hand side, you've got a transmission electron microscope image of a cardiac myocyte, and you see a very, very structured, organized image. You see here these elliptical structures, which are mitochondria, which are the organelles that produce the energy for the cell, and they are lying in neat rows here throughout the myocyte. In between the mitochondria, you've got myofibers, or the muscle fibers, and these are made from cytoskeletal proteins called actin and from motor proteins like myosin, and these form these little fibers in between. And the cytoskeleton is anchored to these so-called Z-lines, which give muscles a striated or striped appearance. And the sliding of the actin and myosin across each other is what makes a muscle contract. On the right-hand side, you've got a fluorescence image of a ventricular myocyte that was stained for a calcium channel. So in green, you see where this calcium channel is present. And again, you see this striated appearance of the myocytes. So how do these muscle cells contract? Here you see a graph that illustrates an action potential. So the electrical signal coming, for example, from the SA node. And on the y-axis, you do see the membrane potential in millivolts. So once an action potential arrives, the membrane depolarizes. And as a consequence, you see an increase in the intracellular calcium concentration. So here on the y-axis, you see the calcium concentration in nanomolar, and at the bottom of the screen, or the x-axis, is the time. What this video here shows are neonatal myocytes isolated from rats, and they are loaded with an indicator that shows you the calcium concentration. So the brighter the image gets, the higher the calcium concentration inside the cells is. And we are stimulating these cells with electrical pulses, and every time we give an electrical pulse, you see an increase in these fluorescence, which is the increase you see here of the calcium concentration, which then goes down to a baseline level again. So you see that every time we stimulate the cells, calcium goes up. And what you can now see is that, again, with a slight delay in time, this causes a contraction of the cell. So on the y-axis here, the length of the cell is shown. And every time you see a calcium increase, you see a contraction of the cell. And the video just shows slight shortenings of the cells, which are happening at the same frequency as the increases of the calcium concentration. So summarize, the contraction happens after an electrical signal gets to the cells, causes a calcium increase, causes a contraction of the cell, which contracts the whole heart because it's really well coordinated throughout the whole muscle. In a human at rest, the heart beats at around 60 beats per minute. In some people it's faster, and some people it's slower. And in some animals, it's very different. So mice at rest have a heartbeat of around 500 beats per minute. So a mouse heart is beating a lot faster than our hearts. And I think we all know that the rate the heart beats changes, for example, with different demand. And Duncan asked me to pose the question, if you can think of anything that can increase the heart rate, could you please type it into the chat box so Duncan can follow up what ideas you have, what would increase somebody's heart rate? because I really love calcium imaging and it's so important for the contraction of the heart, let's look at another few examples of the calcium signals in a heart working well or not working that well. So here you see another movie of cells loaded with a calcium indicator that show a very rhythmical beating. Again, we are stimulating the cells with electrical impulses, and whenever an impulse comes, you see an increase of the fluorescence in the whole cells, and all the cells show simultaneous increases. So it's very well organized and will cause a very rhythmical contraction of the heart. Here you see an example where you do see whole cell increases at certain times when we give the electrical impulses, but in between you see calcium waves moving in the cells. So that has a potential for disrupting the responses of the heart. And it means that you might have disruptions of the rhythmical contraction of the heart. And in the final movie, you only see these spontaneous calcium waves. You don't see any time where all cells respond. And believe me, these cells are receiving the same electrical stimuli as you see in these two videos. So these cells don't respond to the electrical stimulation that they would receive from the SA node, from the cardiac pacemaker. So these cells would not cause a contraction of the heart and would be a very bad situation. And with that, I'm coming to the end of the structure and function of the heart. You've seen that cardiac muscle cells have a very organized structure of myofibers running through, interrupted with Z lines. They contain many mitochondria, which provide the energy for the continuous contraction of the heart. The electrical signals that the SA node produces causes an increase in the calcium concentration, which in turn causes the contraction of cardiac muscle cells, and they provide the rhythmical contraction that you hopefully have throughout your whole life. And the rate of the heartbeat can change upon demand, and that's the moment when I hand over to Duncan.
Thank you, Katja. Okay, so you should be able to see a blank screen at the moment, and this blank screen really is about the things you have told us in the chat box that affect heart rate. And I noticed that some of you have said stress, excitement, fear, caffeine, and these are all valid. And in the other chat box, we have emotions. I've collected some of your answers into collective terms of emotions. So I furiously had to update the slide as you were putting your suggestions in the chat box. Also exercise, and that can take many forms. A variety of drugs. So I've put the caffeine together as a drug, and that will affect heart rate. And relaxation, of course. What the question was really about affecting heart rate. You can decrease heart rate as well as increase heart rate, and I'll be explaining that in a minute. And so now you should know something about the inner workings of the heart. And the heart is perhaps the only organ whose function we are aware of, because it beats, and it's this beat that can be felt on the body surface. And doctors use this to their advantage. We also know that the heartbeat changes in emotion and exercise. And possibly for this reason, many poets and composers have made reference to the heart. For example, in Macbeth, Shakespeare was certainly aware of the link between emotions and the force of the heartbeat. And the reason for this is that the heart reacts to various stimuli by changing its rate of contraction and force of contraction. So, although you've given me some idea of the types of things that affect heart rate, in this next slide, you'll see quite a lot of them. On the left-hand side, you have anxiety. So if you're anxious or stressed, that very often would lead to an increase in heart rate. Exercise has to increase heart rate because exercising muscle is energy demanding. So amongst all these other things, you have walking and running and low temperature. So if you're unfortunate enough to find yourself in cold water, really cold water, i.e. the North Sea in winter, which is about 5 degrees centigrade, after about half an hour, your core temperature may go down to something approaching 34 degrees centigrade, in which case your heart rate will plummet. So low temperature also affects your heart rate. Hormonal changes. I'll be talking about some of the essential hormones that are involved in cardiovascular regulation. But hormone changes quite often, for example, thyroid hormone elevation increases heart rate. Blood glucose level, alcohol, cocaine, amphetamines, drugs like that would also increase heart rate. And conversely, I said that low temperature hypothermia decreases heart rate. Well, when you have a fever or high temperature, very often your heart rate is elevated. What is surprising, though, is that posture and body position affects your heart rate. So tilting someone, you can definitely see a changing heart rate from lying down to standing up. And what is also quite surprising is that all of us have what we call a diving reflex. So that if we were to dive into water, one of the immediate effects would be not only to suppress respiration for obvious reasons, but also the effect of water on the face decreases our heart rate. And this is a reflex which is built into us when we're born. Even babies, for example, if you were to simulate this by blowing on their face, that would decrease their heart rate and stop them breathing. So I don't advise you to do that, but that's something that happens. Emotions, obesity, medications, and so on. So all these affect heart rate. So it's quite a lot of things. The heart is really quite an important organ, and I'll be talking about how we affect heart rate. So in this diagram, these are things that affect the heart, either directly or indirectly. You're not supposed to be able to read the slide. And the reason I put this slide up is that it shows you the huge number of things that affect heart rate in one way or another. So you have, let me just put a pointer on. You have, for example, environmental factors, work-related factors, non-modifiable factors such as age, gender, or ethnicity. You've also got lifestyle factors like support, posture, exercise, meditation, tobacco, and alcohol. And these are all things you have some control over. You also have the physiological and pathological factors. So changes in heart rate can be normal, and also they can be abnormal, depending on whether you've got an infection. And these would include asthma, cardiovascular disease, and the endocrine factors, including hormones such as cortisol. But also you've got central nervous system effects on heart rate, and also neuropsychological factors such as mood, depression, anxiety, emotion, behavior, and personality changes. So let's just go on to the next slide. So you can see a variety of factors that change heart rate. So in this diagram, in this slide, you can see two electrocardiograms or ECG traces. That's the electrical activity of the heart recorded using electrodes between the arms and the legs. These would typically be types of records obtained by a doctor or a nurse. So if, for example, you wanted to have some investigation on your heart, one of the tests they would do would be an ECG, or electrocardiogram. So these are measures of the electrical activity of the heart as the heart undergoes different stages of heartbeat. So at the top of the slide, you just get my laser pointer at the top here, we've got an ECG, and it shows a slow heart rate. So these are depolarizations of the heart. This is depolarization of the atria, the ventricles, and repolarization of the ventricles. So it shows a slow heart rate, as I said, where the peaks of activity are separated by large gaps, and this is called sinus bradycardia. Bradycardia is Latin for slow heart. Whereas at the bottom, we have what's called sinus tachycardia, which is the reverse. So this is a much faster heart rate. This is the same rate of time. They're both over the same time. And you can see at the bottom slide, sinus tachycardia, the heart rate is much faster. And tachycardia means Latin for fast heart. So let me just show you the next slide. So we can measure our own heart rates. We're not normally able to measure our own ECG with smart devices, but this is a smartwatch, and this is my Samsung Gear. It's now 8 years old. It was given to me by Samsung following a project I did with them. I was working on one of their apps that measures alternative forms of exercise other than simply walking or running. That was meant for people over the age of 55. So what I would normally do is to look at the Samsung Health app on my watch, and then you bring up a function looking at how you record from the heart, and you can see in the middle of the screen measure. I would press that, and what would then happen is I would get a trace like this. This is a trace that's called a photoplethysmography trace, or PPG signal. And that's how most of the smartwatches actually derive heart rate. They derive it from this. And this is the pulsatile nature of the blood immediately underneath the strap. And this is where you'll see the two red lights and sometimes a green light showing when you take your watch off. And this is a sort of trace that's happening. I need to turn off my pointer to run a short, very, very short video. So this is what will happen, and then it do a calculation. In this case, the calculation was 76 beats per minute, which is slightly higher than resting heart rate. As Katja said earlier, resting heart rate is about 60, maybe up to 70, and this is slightly elevated. So this is sort of more reasonable for someone at rest. And then in the next slide, I'll show you the way in which the body changes or influences the heart. This shows you the parasympathetic and sympathetic divisions of what we call the autonomic nervous system. And the autonomic nervous system is really a system whereby we have very little control over. It's the way in which our body is maintaining control over the peripheral organs. And you can see here on the left and the right, let me pick up the laser point again, on the left and the right, these are the parasympathetic connections, and on the right, the sympathetic connections. So many of you are likely to be familiar with some of these concepts of sympathetic, parasympathetic, and you will know that the sympathetic nervous system is responsible for the flight or fight reaction, and that occurs in response to a stressful stimulus. And that's one of the questions, or should I say, one of the suggestions by you in the chat box. So let's go into the left-hand side, the parasympathetic nervous system. If you, let me just click again. This is the heart, that little part there. And the parasympathetic nervous system causes a decrease in heart rate when activated, and it does this via the vagus nerve. Here's the vagus nerve. On the right-hand side, we also have a picture of the heart. Obviously not two hearts, but actually trying to divide the two parts of the autonomic system. So in this case, the activation of the sympathetic nervous system is accelerating the heartbeat and increasing the heart rate. So, the sympathetic nervous system increases the heart rate. It does more than that, actually, and I'll be just talking about that in a minute. It's not just the nerves that can change the state and rate of the heart, but also bloodborne substances such as hormones. So this is a diagram showing the innervation of the sinoatrial node. The sinoatrial node is something that Katja mentioned in her part of the talk, the first part. And that's important because it's sort of like where nerves come in from the parasympathetic and sympathetic nervous system and then influence structures in the heart, which then influence other structures in the atria and ventricles. Let's just zoom in. So you can see cells in the SA node here, and the other area is the atrioventricular node. They're two nodes that are electrically excitable. They're waiting for their triggers from the parasympathetic and sympathetic nervous system, and they influence either increase or decrease heart rate, and they do that via their signals to the atria. So you can also see sympathetic nerve fibers here going to the SA node and also to the AV node, but also you can see they innervate or project to the ventricular myocardium. The parasympathetic nerves don't do that. So one of the other functions of the sympathetic nervous system is to increase the force of contraction of the ventricles. So not only can you get, under stressful conditions or exercise, an increase in heart rate, but you can also get an increase in force of contraction, which means more blood is forced out of the heart. So let's just take a look at the list of one of the things that we talked about earlier, hormones. So this is a diagram of the adrenal glands. You've got two adrenal glands, the right gland here and the left one here, and they're just above each kidney. Adrenal means above kidney. And we've got a cutout. We've cut across this section of the adrenal gland, and this is what this figure is here. And you can see you've got a capsule, the adrenal cortex, and the adrenal medulla. I won't explain this diagram here. So the function of the adrenal medulla actually is to release the two hormones, adrenaline and noradrenaline. And once they're released into the circulation, they travel around the body, and apart from affecting all structures in the body, they also have a direct effect on the heart. So once in the bloodstream, these hormones, these two catecholamines, travel around the body and affect the heart. And they do the same thing as sympathetic nervous system activation, so that increasing heart rate and also force of contraction of the heart. So what are the things that affect heart rate? You've seen this diagram before, and I'll just concentrate on the one thing I think which is really quite important to show you, and that's the exercise. That's one of the things that obviously increases heart rate. I'll just show you how it does this. So what is happening in exercise? Well, we know that muscles are being activated. You want to exercise. Type of exercise could be swimming, it could be running. Doesn't really matter. It's an increase in metabolic demand on the body. So your muscles are activated. Well, they are metabolically active. They need to be provided with more nutrients and more oxygen during that process. But also the heart rate increases on exercise. In some athletes and some people, however, the heart rate may increase before you exercise. There's a preparation for exercise, a priming for exercise. So sports physiologists, for example, would know that and measure it in individuals. Also, as I said, the force of heart contraction increases. And the reason for this is that you need to have more blood coming out of the heart. It's all very well increasing heart rate, but if you decrease the size of the ventricles at the end of contraction, you expel more blood. Okay, and that means also that more blood will flow into the heart, because the sympathetics are activating the veins in the body, and your muscles have veins in them but these veins have valves so that when muscle is active, the blood in them is forced into one direction towards the heart. So your what we call the venous return increases, so more blood flows to the heart. So now we've got more blood pumping out of the heart. And so the result of all of this is your cardiac output increases. Now, obviously, it increases by amount that matches your exercise. So if you were undergoing light exercise, the cardiac output would not be as much as if you were running for your life or for the bus, maybe. Also, because you've got an increased metabolic demand in the body from your exercise, respiration will increase. So this leaves us with a question of, so what happens when the heart goes wrong? So I'm going to leave you in the good hands of my colleague, Dan Johnson, who will take over the slideshow.
Okay, thank you for that, Duncan. What I want to do in the last section of this presentation is to basically discuss, indeed, as Duncan was saying, what happens when things go wrong. And I want to start by discussing a syndrome called heart failure. So heart failure comes in many different flavors, let's say, and these are becoming defined differently constantly. So at the moment, we have a different heart failure syndromes called heart failure with reduced ejection fraction, or heart failure with preserved ejection fraction. And in this presentation, I will concentrate on the reduced ejection fraction. And what that means is that the amount of blood going in and coming out is reduced compared to what it would be under normal conditions. And that's because there's a problem with the way the heart reacts and contracts. So it's nothing to do with the stiffness of the tissue itself, as it might be in heart failure with preserved ejection fraction. So already back in the 1960s, as Duncan was previously discussing about noradrenaline and how important noradrenaline is in the rate control of the heart, in the 1960s, a study by Chidze, which was published in a prominent journal, the New England Journal of Medicine, already showed that the levels of noradrenaline in the atrial appendage, so this is a small part of the atria that couldn't be removed in surgery without many problems being caused by that, and it might actually help in some conditions. So they removed the atrial appendage and then looked at the amount of noradrenaline in the different atrial appendages from the different patients. And what you can see from here, hopefully you can see my pointer, is that in control conditions, for example, we had between 1 microgram per gram of tissue of adrenaline, whereas in patients with congestive heart failure, this was greatly reduced. So this was already indicating that there was already some changes in these patients in terms of their noradrenaline sensitivity and the way the noradrenaline was released from the adrenal glands. Let me click on. So since these initial experiments that were carried out, nearly 80 years ago or so now, well, 60 years ago now, it's been shown that there's really a balance between the sympathetic and the parasympathetic systems really play a major role in heart failure. So what would normally happen is if you have a decrease in blood flow, much like when you have an increase in blood flow, but in reverse, is what Duncan was talking about, when you have a decrease in blood flow, the sympathetic activity will be increased to try and compensate for that. And at the same time, something called the baroreflex sensitivity is reduced. In turn, basically this means that the heart readapts itself and it tries to make its systolic and diastolic force change depending on the adrenergic mediated increase. So that basically the heart tries to recompensate for these changes, but ultimately, because of that, it becomes less sensitive to the stimulation. So you can imagine as you give more and more of the noradrenaline or the adrenaline, those receptors that would normally be receptive to that don't feel as sensitive anymore to it anymore. And you need more of it to produce the same effect. So it's a little bit like the boy who cried wolf in many ways. So it ultimately becomes a vicious cycle, meaning what was trying to help the heart in the first place actually makes it become worse in the end, because you get downregulation of certain proteins in the heart, your structure of the cardiac myocytes may also change in terms of where the receptors are located on the membrane. And there's lots of cellular remodeling that may happen in this syndrome because of these differences in the parasympathetic and sympathetic systems that occur as part of this. At the same time, we have beta blockers. So as Duncan mentioned earlier, we have adrenaline, and we have these are normally target these beta adrenergic receptors. So what we have is beta blockers. Some of you may have heard of these before. And these are drugs that actually block beta adrenergic stimulation. So they might slow your heart rate down, for example. But what they do is they remain a mainstay of treatment for heart failure, and also, for example, in arrhythmogenic syndromes such as atrial fibrillation, which maybe some of you have heard of. Okay. Oh, wrong way. There we go. So what I want to talk about now is move a little bit away from heart failure, but also then go back to how the sympathetic system can affect other channels and other syndromes that we see within within cardiology. So as Katja spoke about right at the beginning of this presentation, the heart's pumping function itself is driven by this electrical signal. And this electrical signal, these action potentials, basically occur because of different ion channels on the surface of those cardiac myocytes. And they open and close either due to different substances or due to the voltage, because they're voltage gated, or ligand gated is what they're called when they're gated by a certain substance. However, sometimes these ion channels may contain mutations, which means that actually they react in different ways to how they should. So maybe what was meant to be a channel that closed very quickly takes a little bit longer to close, or they open at a different voltage to where they should have been opening. And obviously, as you can imagine, if you think back to that picture of the action potential that Katja showed right at the beginning, if you start changing the opening and closing of those ion channels, you will also change the shape of your action potential. And there's a group of those cardiac syndromes, which means the electrical beat of the heart lasts longer because of these mutations. And that's known as a number of conditions called long QT syndromes. And there's at least 8 of these now that have been defined. I think there's up to 13, if I remember correctly. But these patients actually are much more likely to have these negative cardiac effects, such as cardiac arrest. And in cardiac arrest, and fibrillation is what it's called. So you have ventricular fibrillation as opposed to atrial fibrillation, when it's only your atria that's contracting erratically, which isn't in theory life-threatening by itself. But when your ventricle fibrillates in that way, it basically means that it's not pumping the blood around effectively enough. And a definition that's always stuck with me from my undergraduate days was what is called a bag of worms. So if you think about a bag full of worms in it, you see them wriggling around. And so it's not contracting completely like this, but it's going a little bit like this. So it's not being able to push around the blood into the aorta and around the body as it should be. And there are multiple things that trigger this. And I'd show this paper again. This is quite an old paper from Circulation, so another prominent journal from the early 90s. And they show in this particular syndrome, so they looked at long QT1, long QT2, and long QT3 here. So long QT1 and 2 are both due to mutations in a certain potassium channel. So long QT1 is to do with the slowly rectifying potassium channel. You don't need to worry about that. Whereas long QT2 is more to do with the rapidly rectifying, so the HERG channel. And you see in those two syndromes that these are much more likely to trigger events when the person is exercising or has an emotional event happening. So they're much less likely to occur in sleep or in rest. And the same is true in terms of sudden cardiac death. You see indeed 68% of those patients, out of those 57 patients who died during that time, died because of at the time of exercise. So this tells us, and we know from other experiments and other research that's been carried out since then, there's a massive link between the sympathetic system and this long QT1. So we know that if we can block, for example, the sympathetics in long QT1, these patients are much more likely to survive. And that's one of the mainstays now of treatment in these patients. that is given to help them. On the other hand, if we look at long QT3, where we have a lot lower amount of patients to start with, so they only managed to recruit 62 patients in this study who had events. You see, actually, these guys are much more likely to have their events, and the ones that die, again, there were 25 of them on here, they're much more likely to die when they're sleeping and when they're resting. So it's a little bit of a reverse of what happens in long QT 1 and 2. So this is less likely to be driven by the sympathetics, but it might be driven by either the parasympathetics or maybe not driven by any of those at all. And we know, for example, that long QT3 is a mutation in a sodium channel, and that sodium channel is normally involved at the beginning of the action potential. So that opens right at the beginning to cause that initial depolarization. And what happens sometimes is that stays open for longer than it should do. And that's what causes this long QT3 syndrome, because that also changes the amount of calcium entering the cell and the length of the action potential during that time. But this doesn't seem to be affected by adrenergic stimulation. So it would be a very bad idea to give long QT3 patients, for example, a beta blocker, because that might make it actually worse, because in those situations, the adrenaline that the patients might be receiving might be helping those guys stay alive. So it's important also to define which syndrome the different patients are suffering from. And this is also where genetic screening becomes very important, to be able to understand exactly what genetic mutations these patients may have. So what's also been done, looking at adrenaline in the clinic, it's also been used as a masking event. So when thinking about diagnosing patients, what's happened in some papers, so in some clinics, this was from now a bit more recent, from 2018, what these guys did was they basically gave a number of patients a mental stress test. So I think from what I remember correctly, it was an arithmetic test, and they had to do it in a quick amount of time. And whilst they were doing that, at the same time, they were recording the ECG. And from the ECG, you can then record your QT time. And if you remember from what Duncan was showing, you have your QRS complex, which is the depolarization of the ventricle, and the end of your T wave, which is your repolarization of your ventricle. So if you look at your QT, that's a surrogate marker for the action potential duration. But obviously, it's not that simple, because they are looking across the whole heart and not just at a single cell, for example. But what's been shown is that these two are quite linked. So what they did in these patients was see what happened under different conditions when they gave stress to these patients, to see what happened to the QT, if it prolonged or if it shortened. And if it prolonged, for example, that might have been a hint and a little bit of a diagnostic test, which would mean further screening to go on to show that these patients might have had long QT1 or long QT2, so a syndrome that was sensitive to these catecholamines, these adrenaline and noradrenaline hormones in the first place. Again, also, as we've said, because this is genetic, it's also important then to think about testing relatives, so parents or children of patients who come into the clinic. And that's also been done in terms of exercising patients and then looking at to see how that links up with long QT in those patients and how that fits with the genetic testing that might also go on. So these have all become quite important over the years in terms of defining and diagnosing these different syndromes. So what I want to end with talking about is this final syndrome, which is broken heart syndrome. So this is actually, it's called broken heart syndrome, but it's the actual proper name is Takotsubo cardiomyopathy. Cardio meaning heart, and myopathy meaning the muscle and muscle growth. So what we have here is a syndrome which happens when basically the heart muscle suddenly becomes stunned. And this was defined back in 1990 in a Japanese patient. And that's why it got named at that point, because Takotsubo actually means an octopus pot. And I'll come on to show you why that is in a short time. So we don't quite know what happens in these patients. We know that the majority of patients come and show and present themselves as being postmenopausal females. So there might be some hormonal component to this, or maybe premenopausal, those hormones before menopause have a protective effect in women. And it looks like in patients who have this syndrome that they are having a heart attack, otherwise known as a myocardial infarction. So they would present to the clinic being out of breath, having chest pain, and maybe also fainting ultimately. So at least a third of these patients appear to be linked with an emotional trigger. So that could be for many reasons. As you can see, I've shown on the left here a diagram showing most of those things, including emotional and physical triggers. So emotional triggers could be things like illness, death of a loved one, job loss, a car accident. Even when people haven't been physically injured, the stress emerging from that car accident has also been shown to cause this broken heart syndrome. Again, there's all these different things that can lead to this increase in catecholamines and adrenaline that is thought to be one of the major drivers here. At the same time as what might be seen as these negative triggers, happy triggers and positive triggers might also trigger this. So I think there's been at least one case showing this has occurred after somebody has had a lottery win, for example. So you've just got to be a little bit careful and make sure you control a little bit your emotions to make sure you don't get an overriding of adrenaline or noradrenaline in this situation. At the same time, there's different physical triggers. So for example, surgery, chemotherapy, even when your COPD, so chronic obstructive pulmonary disorder, gets worse, has also been shown to cause this as well in terms of physicality. Also, interestingly, administration of catecholamines, which might be done in the clinic, has also been shown to do that. So sometimes you might give adrenaline to a patient, for example, for some reason, and that has been shown to induce this syndrome. And if we look at what actually happens in this situation, here on the left, we have our normal heart. So here, I'm looking specifically here at the left ventricle. So as Katja said right at the beginning, the left ventricle has a very thick muscular wall because this guy has to pump the blood into the aorta, which is this major blood vessel here, the major artery, the major, let's call it the M1 if you're in the UK, of the body's vascular system. And basically this then goes off to all these different A roads, let's say, to go to the rest of the body and to the rest to oxygenate and take away all the bad stuff back into the blood as well. And what you see here is a thickened wall, because obviously you need to pump quite hard to be able to get that force going. And here you have the actual blood-filled cavity, the blood-filled ventricle. If you look in this syndrome, so in Takotsubo cardiomyopathy, I will get that right at some point, what you see is there's a very different shape. So if you look here, you have a much thinned wall of the left ventricle, and you have this kind of shape here, so where it, divots in a little bit. So there's a contraction here. And if you look here, this is what these octopus pots look like. And therefore you can see why it was called this in the first place, because it kind of resembles this kind of octopus pot. And if you think about what might happen because of this, this means obviously, if you don't have as much muscle mass pumping the blood around, you probably won't get the same force, so you won't manage to get the blood to everywhere you want to do. At the same time, you also have a shortened and a narrower entrance into the aorta, which will obviously also make it more difficult for the blood to get into the aorta to get going in the first place. So, and because of that, you will obviously have the symptoms that I spoke about previously, and because of the fact you have lack of oxygen in different areas. Some mechanisms, so we think again about stress and the adrenergic system. So that stress is caused by a trigger, which then might target the limbic system in the brain and hits the hypothalamus and the locus coeruleus. Don't worry about those names, but they're just certain areas of the brain that are known to be involved in stress mechanisms. They cause release, they cause activation at the ganglia, and cause neurally released catecholamines, which might then drive the syndrome. At the same time, they'll hit the adrenal glands and the pituitary glands, which will ultimately lead to these adrenal catecholamines, as Duncan was speaking about before, which will also drive this syndrome at the end. So it's likely that this occurs because different regions of the heart, so for example, the top of the ventricle versus the bottom of the ventricle, might have different sensitivities to the different compounds and the different catecholamines that we've previously discussed. There's still a lot of research needed into this, however, and we're doing some of that at the OU, not specifically into this syndrome, but into what happens and how these different sensitivities are across the whole surface of the heart. There's also been other causes, and I thought these were quite interesting little case reports to share with you. So, for example, back in 2012, there was a patient who had anaphylaxis and then presented with this Takotsubo cardiomyopathy by eating these sago worms. And that obviously caused some release of catecholamines, which led to the patient ultimately getting this cardiomyopathy. At the same time, there was another patient back in 2019 who got triggered cardiomyopathy because they started eating wasabi. And if you like your sushi, you know that wasabi is this green substance that you eat together with your sushi, but it can be quite strong. And they thought it was avocado. So when they took that in the same amounts as avocado, obviously it's going to have a very different effect to avocado, and it ultimately triggered a massive surge in these catecholamines, and they ended up getting this cardiomyopathy at the end. A final case that I want to discuss with you, this was from a couple of years ago. Hopefully this video will play. Let me just get that working. Oh. To do that. No, okay, it's not working. The video's not working for some reason. But what it basically was showing was this 47-year-old appeared in the clinic, and she'd had vivid hallucinations of being chased by men and taken as a prisoner, as she's just been reading one of the Harry Potter books, and I guess it was Escape from Azkaban. And what they showed in this patient was that they ended up with Takotsubo cardiomyopathy. And let me see if I can get this, because it's actually quite a nice video to illustrate. There we go. Hopefully there we go. So that's running now. Now you can see what's going on here. So if you look now, hopefully you can see my pointer now. Here you see an MRI scan. So you have the left atria here, the left ventricle, the right ventricle even. And this is the left ventricle. And if you look at the beating of the left ventricle, you can see that it beats. And at the end of the contraction, it really does look like that octopus pot as we showed before. And what you also see by looking at this, at the bottom of the heart, there's not much muscle mass here. So obviously that's not going to contract as much as it should be doing to get the blood to the rest of the body. And therefore we have problems. Good news here is most of this syndrome is reversible. So most of these patients recover, but it's just something to be aware of in this from these different aspects. And then basically in this situation, it was thought this cardiomyopathy was because of this sympathetic autonomic surge from these vivid themed hallucinations. So I thought that was an interesting case study. So in summary, so to sum up everything that we've been discussing, the pumping action of the heart is carried out by the cardiac myocyte. These aren't the majority of cells in the heart. However, they do take up the most volume of the heart. These guys have specific structures and proteins that allow them to behave in the way they do and allow them to contract. Calcium, so one of the ions that we are looking at in various diseases, but also in cardiac diseases at the OU, is a key regulator of cardiac contractility. The heart rate and intensity of the contraction is under careful control of a number of different pathways, as Duncan was showing us in the middle of the presentation, including the sympathetic and the parasympathetic nervous systems. However, sometimes we've got to think the careful regulation of these processes can become unbalanced, and this can lead to various heart problems, as I've discussed a few of them in the last part of this talk. And we need to have continued research of these diseases and the rest of the heart diseases and these mechanisms to hopefully lead ultimately to better diagnosis and treatment of these diseases. And we're carrying out that research, some of that research at the OU, and collaborating with various people around the world to do that. So hopefully, ultimately, we will have a better treatment and a better handle on what is going on in these different conditions. And with that, we will end. And if you have any questions, please put them in the chat area on Facebook or YouTube. And we hope you enjoyed this session.
Thank you, Dan. We do have quite a number of questions. So the first one, I don't know who wants to answer this. What is cardiac output?
You go for it, Duncan.
Oh, right. Okay. So cardiac output essentially is a measure of how much blood is coming out of the heart in a minute. Okay, so it's the stroke volume, i.e. a heartbeat's worth of output from the heart times your heart rate. So at rest, this will be something along the lines of 5 liters per minute. And in maximum exercise, it might be as much as 25 liters per minute. So in other words, 5 times the amount of blood going through the heart and coming out at the other end. Another question was, how common is sudden cardiac death or the syndrome?
I don't know the actual numbers of that, but I should do, because it's also difficult to know who's died of sudden cardiac death because not everybody has an ECG on all the time and a Holter monitor, so you can't always define who has died from sudden cardiac death. Although you do have to be careful, because you see in the press and various websites, you think people discuss sudden cardiac death and heart attacks as the same things, but they're very different things. So sudden cardiac death is actually because of the electrical activity of the heart ceasing to work anymore or not operating correctly anymore. Whereas a heart attack or myocardial infarction, as we discussed earlier, as I showed earlier, is actually when the blood supply to the heart itself isn't getting there anymore. And then ultimately that might lead to arrhythmia and sudden cardiac death, but they're not one and the same thing. So that's always something where you have to be careful about. And it's very difficult to define who had sudden cardiac death. So you might remember in the Euros of the World Cup, we had Eriksson from the Danish team. He had a sudden cardiac event, it looked like. So he probably had this, like what's called a torsade de points. I won't go into details about that, but it basically means turning of the points, arrhythmia. Arrhythmia meaning lack of rhythm. And that's why in that moment of time, he collapsed during that moment. And there's been a number of different occurrences of that happening on the football field, if you look back in history.
Yeah.
So it's
gone onto the British Heart Foundation website. It's about 500 people a year.
Okay.
On this particular thing. So post-mortem, there's no indication of what the problem was. They have to be reasonably young and fit.
But you can also have sudden cardiac death in older people as well. So it can, and it also might be one of the occurrences of sudden infant death syndrome. Yeah. It's again something we don't really understand that much about. But some patients, when they've managed to look at sudden infant death syndrome and looked at the genetics afterwards, they've been shown to be carriers of certain mutations that would make these patients more liable to get a cardiac arrest.
And the last one is for you, is what is QT? You mentioned QT quite a lot.
Sorry, I thought, so we spoke, so basically...
Do you want me to put the figure of the...
If you put the ECG back up, Duncan.
Yeah, okay.
Yes. So the QT is basically something that's measured on an ECG. And if that picture comes back up... Okay, maybe I can get my pointer. Oh, can I get my... Oh, I can't get my pointer up. So maybe Duncan, you can point to the, yeah, exactly. Go back to the bottom. So there you see, that's the beginning of the Q wave. So that's the beginning, the QRS complex even. So you have the QRS complex, QRS, and that's basically your ventricular repolarization across the whole of the heart. Then you have your T wave, which Duncan's going to now, which is the ventricular repolarization. So then if you measure from the beginning of the Q to the end of the T, Duncan, yeah, that's the QT interval. And that basically will give you some idea of how long it takes to depolarize and repolarize the whole of the ventricle. And sometimes, as I said, that's prolonged because of these changes in these ion channels and mutations in these ion channels that we have. Okay, I hope that makes some sense.
It's past now.
Okay. So thank you everyone for coming along this afternoon and using your lunch break. We hope you've learned something about the heart and the functioning of the heart and what can go wrong. And with that, I would also like to thank my co-presenters, Katja and Duncan, and also people behind the scenes who've been looking after all these streams today.